Hey guys, how's it going? It's Dr. Weaver here with another study tip for the topic in genetics for AP biology based on the kind of questions that they asked before. Last video, we talked about the gene expression in prokaryotes in the operon system. Here with this, we're going to talk about eukaryotes. There's a couple of similarities. One is going to be a gene is going to be under control, whether it's going to be turned on, which means that the DNA is going to be expressed into messenger RNA, or it's going to be turned off or silent. RNA polymerase is going to want to bind to this promoter, which is right in front of the gene. This way it could zip along and transcribe the gene. Now we could think of this as a river, as I said before, where upstream is before the gene, is downstream is going to be towards the gene over here. So upstream in the eukaryotic system, we have these things called enhancers. Now these enhancers are going to be binding elements for things like activator proteins are going to come bind there. And activator proteins are also known as switches. So in different parts of the body, you could actually have different switches or different activators that are going to cause the promotion of this gene being expressed. You're also going to have a clump of proteins called these mediator proteins, and you're also going to have these factors or proteins called transcription factors. Transcription factors, mediator proteins are all going to come together in the presence of RNA polymerase only when activated proteins are going to be bound to the enhancers. So it looks something like this. The activator proteins are going to bind to the enhancers. What's going to happen is the mediator proteins are going to come into the region. Transcription factors are going to also be in the region. They're going to assemble at the perfect conformation, and that means RNA polymerase is going to go down the line and transcribe the gene, and then you will ultimately translate the protein as long as everything else is in place. Okay, a quick observation is that this DNA actually bends for this conformation. Doesn't it look like a U? Well, here's a study tip for you. Whenever you see this diagram, which has shown up before, that it looks like a U, and it is not to do with a prokaryotic gene expression, but a eukaryotic gene expression. So here's the U right there. The other study tip is that stickleback fish have found their way on the exam at least a couple of times. And with the classic experiment and video by HHMI, they show the stickleback fish have lost their pelvic fin. Now, in the case of the stickleback fish, what happens is each of these enhancer regions with the activator proteins acts as a molecular switch. Rather than having the mutation in the gene that is going to make the actual fin or the limb, there's a mutation in the switch. So you may actually have one switch that is going to allow the expression of this gene in the pituitary region. You may have another enhancer region with a switch that is going to allow the expression in the jaw region. And you may have a third region over here that's in the pelvic region. So in the case of the stickleback fish, there was an actual mutation in the mechanism of the switch for the pelvic region, thereby not allowing the expression of this gene in the pelvic region, yet still maintaining it in the jaw and the pituitary gland, so there was not a total loss of the gene. I don't know if you guys noticed, but right over here, there's a link towards this HHMI video on the stickleback fish of exactly what we talked about at the end with these switches. If you haven't seen it before, I suggest that you check it out right now. And just as a quick note, I don't know if you guys have noticed in the past, but if you've seen a lot of these HHMI videos and done a lot of the practice AP exams, that there is a definite correlation between the material covered in these videos and series of questions that occur on the exams. So I would explore the site and check it out if I were you. That is my final study tip for genetics. Good luck, guys.